Okay, here's the title again. I will be talking about a new technology called RNAscope, which is really a ultrasensitive RNA in situ hybridization technique. I will demonstrate how this new technology can be used to overcome some of the key challenges in cancer biomarker research. First, in the RNAscope technology. So RNA in situ hybridization is not new. It's actually a technique has been used for over 40 years in molecular biology as a powerful tool to localize a specific nucleic acid sequence to a specific cell or subcellular structure without isolating or extracting the RNA. Initially, it started out with using radioactive label probes. Later on, non-radioactive methods using fluorescent label probes or chromogenic detection methods were developed. However, non-radioactive detection systems are not, in general, are not as sensitive as radioactive label probes. So there are a lot of efforts were made to improve the sensitivity of RNA in situ hybridization. This includes in situ PCR, where you amplify your target by PCR before you perform in situ hybridization. You also have various signal amplification methods such as TSA, branch DNA, rolling circle amplification, hybridization chain reaction that amplifies the signal after hybridization. However, despite all these improvements in RNA-ISH, it remains to lack sufficient sensitivity and specificity for regular routine use, especially in a clinical setting. So that's why ACD developed a new technology called RNAscope in 2010 to address these challenges. So what's different between RNAscope and conventional RNA-ISH? Well, first, all the previous improvement in RNA-ISH have focused on increasing sensitivity without addressing background issue. RNAscope is designed to increase sensitivity and suppress background at the same time, giving you a net increase in signal-to-noise ratio by 400-fold. The RNAscope assay is also extremely streamlined. It can be run in a single day compared to a multi-day procedure by conventional methods. So here is a schematic of the RNAscope assay. Basically, it looks pretty much like conventional assays in that it consists of the same steps of a cell permeabilization and hybridization with the target probe and signal amplification, and then visualize your signal under a microscope. You may notice here that the target probe for RNAscope seems to be unique, as is depicted here as something like letter Z, which actually is the basis for us to achieve high sensitivity and very low background. How the probe design works explained here. With the probe design, we call the double Z probe design. Each Z has 3 regions of sequences. On the lower part of the Z is an 18 to 25 base sequence that is complementary to a target. In the upper region of the Z is a 14 base tail sequence that is complementary to the signal amplification system. All the Z probes are designed to hybridize in pairs. That is, they will hybridize in tandem to a target RNA to a region about 40 to 50 bases. At the same time, the double Z pair also forms a 28 base region that is the pre-amplifier binding site. We typically design 20 double Z pairs across a 1 kb region of RNA. The 2 things I want to point out, one is that the relatively short target region of a double Z pair, that is a 40 to 50 base pair, really makes the assay compatible with a partially degraded RNA, as often the case in clinical specimens. And the other thing is that we designed 20 pairs of double Z probes for target, even though we have shown that only 3 pairs of double Z is required to visualize the signal. So the 20 pairs offers redundancy and also robustness for the assay. So how signal amplification is achieved is illustrated in this animation. And again, this is a target RNA transcript shown on the bottom. The double Z probe binds to the target, which forms a binding site for the pre-amplifier. And then the amplifiers bind to the pre-amplifier, and label probe binds to the amplifiers. With the 20 double Z probe pairs, we can build a tree like this to accumulate as many as 8,000 label molecules per target RNA. So that is the basis of the high sensitivity of the RNAscope technology. Sensitivity is only a part of the features of the technology. We also address background, and this is illustrated here. In the case of a non-specific target, one would expect only one of the double Z pair may be able to bind to this non-specific target. However, this single Z binding does not form the 28 base pair pre-amplifier binding site that allows the stable binding to the pre-amplifier. So a single Z binding to a non-specific target will not support signal amplification. This design concept is validated in this experiment. So we designed the probes against ribosomal 18S ribosomal RNA, and then used a signal amplification system with 16 amplifiers, and compare that to a system that don't have 1 amplifier. It means there's no amplification. Look at panel B, as you can see here, with no amplification. You can see a faint background of fluorescence in the cytoplasm, which corresponds to the 18S ribosomal RNA. With the 16S amplifiers, you see a significant increase in fluorescent intensity, and a quantitation of these 2 images indicates that the increase in signal is quantitative. That is almost 16-fold. To demonstrate that the double Z binding, that is the simultaneous binding of both Z probes, is required to generate signal, we use one of the Z probes, either the one on the left or the one on the right, to hybridize to the cells. As you can see here, no signal is detected by the single Z probes. And here is the 18S sense probe serving as negative control. This shows that the double Z probe binding is required for signal amplification. Hence, that's the basis for background suppression. The double Z probe design enables readily detecting multiple RNA species. In fact, you can do this in 2 different ways. One way is that with multiple targets, you can design probes, each that have a different tail sequences recognized by different pre-amplifiers and label probes, which give you discrete signals for each target. With the use of fluorescent probes, we can currently achieve 4 targets detection. Second way of detecting multiple species is by pulling the probes for multiple targets. This can be designed by attaching the same tail sequences for the different targets, so that all target probes are recognized by the same pre-amplifiers and label probes, which generates a pooled signal. And this approach can be quite useful. For example, in the case of HPV, there are more than 10 different subtypes you want to detect. You don't care which one you are detecting, and you can combine all the probes for the different subtypes together and detect the signals as a pooled signal. This is an example of a 4-plex RNAscope fluorescent assay. The 4 targets are being detected. Red is the beta-actin and HPRT1, RPLP0, PPIB. The blue is the DAPI stain for the nuclear DNA. As expected, the red beta-actin signal is present throughout the cytoplasm, almost throughout the cytoplasm. And on the other hand, the 3 other genes, the green, the HPRT1, RPLP0, and PPIB, the signals appear as punctate individual dots. This experiment compares the performance of a chromogenic RNAscope against isotopic in situ hybridization in detecting IGF-BP3 expression in Kupffer cells in the liver tissue. In the chromogenic RNAscope assay, specific intense staining of a subset of cells are easily visualized here, and which correspond to the Kupffer cells. And notice the almost complete absence of any background in other cells. In contrast, with the isotopic ISH, even though you could see a concentration of signals around a subset of cells, you also see significant background staining across the slide. In addition, this amount of signal were generated after 48 hours of radiographic exposure compared to a 10-minute chromogenic reaction for this chromogenic assay. So the RNAscope assay generates more signal, much less background, and with much faster detection. This experiment compares a chromogenic RNAscope assay against a state-of-the-art non-isotopic ISH. And this experiment was done by an expert in non-conventional ISH, Jake Estes at NCI. He uses ISH routinely to detect SIV. With the RNAscope assay, the SIV signal appeared intense and highly specific, can be visualized under 5x magnification. In contrast, using his own non-isotopic ISH, the signal is barely visible under 5x. You have to go to high power in order to confirm the presence of signal. So this really shows that RNAscope assay can be as much as 100-fold more sensitive than conventional ISH. The RNAscope assay can be run manually, as shown here. All the steps, polymerization, hybridization, amplification, and washing, all can be done within 8-hour procedure, and the results can be visualized in the same day you do the experiment. The workflow has been automated on existing instruments from Ventana and the Leica. And here, the first 3 steps are fully automated, so you can load your slides onto the machine and push the button. After 8 or 9 hours, your slides are ready for visualization under a microscope, and image can be captured for quantification. As I mentioned earlier, the RNAscope signals can be visualized as discrete punctate dots, and each dot represents a signal from a single RNA molecule. So this presents a simple approach to quantify the amount of RNA at single cell level. This concept is demonstrated in this experiment. We quantified HER2 mRNA in HeLa cells using, as shown here in green, and we also stained the 18S ribosomal RNA in red to show the cytoplasm. We counted number of green dots for about 100 cells, and then come up with an average about 14 dots per cell as an estimate of number of HER2 mRNA copies in a cell. The same cell culture was also analyzed using conventional grind and bind methods, in this case using a Quantigen 2 assay, which detects the mRNA in the lysate. By using a standard curve, we're able to determine that in these HeLa cells, there are about 17 copies of HER2 mRNA per cell. So these two numbers come in very close agreement, indicating that simply counting the number of signal dots in the RNAscope string slide can be a reliable way of quantifying your target RNA at a single cell level. Counting signal dots by hand is obviously very tedious and also be error-prone. So we have worked with experts in image analysis, specifically the company Definiens, to develop a software we call Spot Studio. This is a screenshot of the software. In the middle of the picture here is an image of an RNAscope string slide, and the user has drawn an area of interest as indicated by the red outline. And the software goes in to find all the cells in this region and also identify the signal dots. And then outputs the number of dots for each of the cells identified in this region and show them in a table here. This data can be visualized quickly in the bar graph shown here, which quickly give you a distribution on of the signal dots per cell in the region of your interest. We have used this automated quantification approach to measure HER2 mRNA in breast cancer and compare the results to quantitative RT-PCR. As you can see here, the two measurements are strongly correlated. In particular, the HER2 status is well separated by the RNAscope quantification. You can see here is the other HER2 positive cases, and here are the HER2 negative cases. And this data here is part of a publication that we published in the Journal of Molecular Diagnostics last year, which demonstrated that RNAscope HER2 mRNA assay can reliably determine HER2 status, which actually performed better than qRT-PCR. The RNAscope assay is essentially universal, can be applied to any gene, any genome, and as of July this year, we have achieved over 4,000 target probes in our catalog, which covers a wide range of species, including humans, animals, and zebrafish, and also includes probes for mRNA and non-coding RNA. Any probes not on the catalog can be synthesized, designed, and synthesized within 2 weeks. The assay is, once it's designed, can be applied to any tissues. And here is an example of detecting the housekeeping gene PPIB in mouse. As you can see here, a specific staining of PPIB is detected in all tissues. So in summary, the RNAscope technology has single molecule sensitivity and this is the most sensitive RNA signal detection platform on the market. It has a very high specificity because of background suppression mechanism is built in each probe. The single molecule sensitivity and single cell resolution allows a single cell quantification, as I mentioned, demonstrated for HER2 mRNA. The assay is universal in that it can be applied to any gene, any species, any tissue. The assay also works in multiple sample types, including FLPEs, which is the most common clinical specimen, and fixed or fresh frozen tissues. The assay, like I said, the new probe can be designed and manufactured within 2 weeks, and the assay can be run in multiple configurations, different color choices, brown, red, duplex, or even higher plex using fluorescence. The assays can be run either manually or on automated system on Leica or Ventana. So this concludes the first part of the technology introduction. If you have any questions, please type your questions into the chat box. Open that. Are there any questions for this part of the talk? Okay, so looks like there's no question yet. So I'm going to proceed to the second part of the talk. Okay. Okay, so here, in the second part of the talk, I will use publications that actually appeared in the last couple of years by the users of RNAscope that demonstrate how this technology can be used in cancer biomarker research to address some of the challenges. There are at least two key challenges in cancer biomarker research. And the first one is that tumors are highly complex tissues. They're not just a mass of tumor cells. As schematically shown here, a tumor tissue contains multiple cell types in the tumor microenvironment, in addition to the tumor cells. These cells actually play an important role in supporting the tumor cell growth and progression. So if you have analyzed this tissue tumor by a grind and bind methods and identified a marker, an RNA marker, to be differentially expressed, for example, it would be impossible for you to map back where the signal come from. That is, where the marker you identified are expressed. In addition, when the tissue, the tumor tissue you analyze may also carry some adjacent normal tissue, which may also confound your interpretation if you use grind and bind methods for analyze the marker. The second challenge is in intratumoral heterogeneity. The tumors are highly heterogeneous between patients, but even in the same patient, a single tumor can be also highly heterogeneous. As schematically shown here, this tumor actually contains multiple subclones. Again, if you have used grind bind methods such as RT-PCR to profile a tumor like this, you will not be able to tell there's underlying heterogeneity. So the challenge in cancer biomarker studies is that you really need contextual information to localize where the signal you measured come from, and not just getting a number using assays such as quantitative PCR. This first example of publication by RNAscope user is discussed here. The authors have previously demonstrated that HER3 is constitutively activated in head and neck squamous cell carcinoma-derived cell lines by high-level overexpression of Heregulin, the ligand, by autocrine signaling mechanism. And this data is important because there is a clinical trial suggesting that response to a HER3 antibody may be associated with high level of HRG expression. So the question is, does high HRG expression define a subpopulation of head and neck squamous cell carcinoma patients that has activated HER3, which may be targetable by anti-HER3 therapies? The authors used the RNAscope assay to analyze how Heregulin signals in head and neck squamous cell carcinomas. This is a figure from the paper, and in this study, they have used the duplex chromogenic assay using the red color to detect HER3 and green color to detect Heregulin. The panel A and B shows the examples of staining a normal epithelium. As you can see here, there appears to be a separation of the two colors. That the green HRG staining cells appears mainly in the basal layer, whereas the red staining HER3 expressing cells appears in the squamous epithelium. In panel C is a staining example of a well-differentiated squamous cell carcinoma. And here, they also see a similar separation of the green staining from the red staining. The green signals, which means the HRG expression, is present mostly in the basal layers, basal epithelial cells, whereas the HER3 expression is mainly present in the central part of the tumor. So in both normal epithelium and this well-differentiated example, it appears that the Heregulin signals primarily through a paracrine mechanism. In contrast, in a poorly differentiated carcinoma, you do not see the separated staining of red and green cells. Instead, almost all cells stains positive for both markers. This would suggest that in this type of tumors, the Heregulin signals through a autocrine signaling mechanisms. So by the use of RNAscope, the authors were able to reveal the two subtypes of head and neck squamous cell carcinomas with different modes of Heregulin signaling. This type of subtyping information would not have been possible if a blind buying method was used to analyze the whole tissue. This second example studies hedgehog signaling in gliomas. As you all know, hedgehog signaling is a very important pathway that is activated in multiple cancers, including gliomas, colon, lung, pancreatic cancers, and is a validated drug target. So the authors in this paper ask, in the gliomas, what are the cells that generate the hedgehog signal, and what are the cells that respond to the signal? The authors combined antibody-based multiple immunofluorescence assay with RNAscope assay to evaluate how hedgehog signals in gliomas. One of the reasons the authors use RNAscope is that antibody against GLI1, which is a target of hedgehog signaling, was faint by immunohistochemistry and was undetectable by immunofluorescence. So this is a figure from the paper showing the results of a multiplex immunofluorescence. Since it is difficult to identify the tumor cells in gliomas by morphology, so they use the antibody against IDH2, a mutant IDH2, to mark the tumor cells, as shown here in red. So the first row is a duplex assay against IDH2 and Sun hedgehog. The Sun hedgehog is detected in green. And when you superimpose these two images, you can see the Sun hedgehog expression cell is one of the tumor cells marked by IDH2. So this indicates that a subset of tumor cells expresses Sun hedgehog. Likewise, the receptor for Sun hedgehog, patch1, is also expressed in a subset of the tumor cells. It's indicated in the second row. If you do a duplex immunofluorescence for hedgehog and patch1, you can see that these the cells expressing patch1 and Sun hedgehog, Sun hedgehog signal is in green, and patch1 is in red. These two signals do not overlap. That means that the cells which are expressing the ligand and the cells expressing the receptor are different cells. And in the bottom row is a triple immunofluorescence for IDH2, which mark the tumor cells, and hedgehog and patch1. As you can see here, the Sun hedgehog expression cells and the patch1 expression cells are distinct cell populations, and they're all tumor cells. They're all subpopulation of tumor cells. And then the authors use the RNAscope to further study GLI1. As I mentioned earlier, the GLI1 is a target of the hedgehog signaling pathway, and antibody against GLI1 has only a faint staining by immunohistochemistry. So in panel A is a duplex chromogenic RNAscope assay for Sun hedgehog in red and GLI1 in green. As you can see here, for the most part, the green staining cells and red staining cells are distinct cell populations. And this is similar to what was shown in the previous immunofluorescence. And likewise, with immunohistochemistry, the hedgehog staining, which is the brown staining cells, and the GLI1 staining, which is in purple, are also in different cell populations. This is consistent with RNAscope results. Finally, the authors combined RNAscope and IHC in a single experiment. They stained for the hedgehog using RNAscope in red, and stained for patch1 with IHC in brown. Again, the hedgehog, the signals expressing cells are distinct from cells that stain positive for patch1 protein. So all of this data suggests a novel paracrine signaling mechanism in glioma. That is, there are two different populations of tumor cells. One population of cells generates the hedgehog's ligand signal, and another population of cells actually receives the signal and generates a response. And this mode of signaling is quite different from what has been found in other cancers. For example, in lung cancer, it has been found that hedgehog signals through an autocrine mechanism. In colon cancer, there is a paracrine signaling mechanism, but however there, the signal is between cancer cells and stromal cells. This is the third publication I'm going to discuss. This paper uses the RNAscope assay to develop a new diagnostic assay for FGF23. Phosphoturate mesenchymal tumors of the mixed connective tissue type, or PMT, are very rare tumors of the bone and soft tissues. And most PMT patients have long-standing osteomalacia due to the overproduction of FGF23, which is a hormone that inhibits phosphate reuptake within the renal proximal tubule. So this makes FGF23 a useful biomarker for diagnosing PMT. The question is which assay to use. The antibodies against FGF23 has been shown to be unreliable. So the authors have previously developed an RT-PCR-based assay for FGF23 mRNA. Although largely successful, this RT-PCR assay does have a low specificity due to a lack of morphology correlation and false positives by non-tumor tissues contamination. These are some examples of staining by RNAscope. And here, panel A and B are H&E images of soft tissue PMT, and the left is a low power image, and on the right is a high power image of the same tissue. And this tissue was stained strongly positive for FGF23 using RNAscope. Here, panel D is an image of a bone PMT. As you actually can see here, a piece of bone in this tissue. Again, the RNAscope demonstrated strong FGF23 expression in this bone PMT sample. In this study, the authors were able to show that 22 out of 23 of the PMTs showed strong FGF23 mRNA expression by RNAscope, thus demonstrating a 96% sensitivity. In contrast, in non-PMTs, the specimens they studied, FGF23 mRNA was not detected in the tumor cells or lesional cells. And there were a few cases that showed some positive staining, but the positive staining was in non-tumor cells. And these are the type of samples that will give false positive results in RT-PCR-based assay. So using this novel assay, they're able to demonstrate 96% sensitivity, 100% specificity for detecting FGF23 mRNA. This is the last paper. FGFR1 gene has been known to be amplified in lung cancer, especially in the squamous cell carcinoma, and where you have 10 to 20% of the cancers have amplified FGFR1 gene. And this is in contrast to the case of adenocarcinomas, where the amplification is rare. It occurs only 1 or 3%. It has been shown that FGFR1 gene amplification is a biomarker for predicting response to FGFR inhibitors. However, the authors of this paper asked, can the FGFR1 mRNA or protein be a better biomarker than gene amplification? The approach they took is to use RNAscope to assess FGFR1 mRNA in a large series of lung cancer cell lines and primary lung cancer in the form of tissue microarrays. And here is the results. In panel A are the examples of staining of RNA for FGFR1 mRNA and copy number. And the first column are a positive FGFR1 positive case, where it shows strong overexpression of the mRNA and also a high increased gene copy number. On the right-hand side is a FGF1 negative case where you do not have any signal for FGF1 mRNA and also have a normal gene copy number. However, the correspondence between RNA and gene copy number is far from complete, as shown in the data below. In this cohort, about 151 lung cancers, there were 13 cases were found to have high gene copy numbers, whereas there were 44 cases to have strong mRNA overexpression. There's only 6 cases in common between the two methods. If you look at the adenocarcinomas separately, there are 48 of these cases in this cohort, and mRNA was strongly positive for 13 cases, whereas none of these are positive by the gene copy numbers. This indicates that there are cases that the mRNA overexpression is not related to gene amplification, and also there are cases that have gene amplification but do not have associated mRNA overexpression. And it also shows that the RNAscope assay identified significantly more FGFR1 positive tumors. The question is, does FGFR1 mRNA overexpression predict sensitivity to anti-FGFR1 drugs? And this question is addressed in this study. The authors analyzed 47 lung cancer cell lines for their sensitivity against a pan-FGFR inhibitor, panatinib. And the cell lines are ordered from low IC50 to high IC50 values on the x-axis. So on the left are the cell lines which are highly sensitive to the drug. On the right-hand side are the cell lines which are highly resistant to the drug. FGFR1 gene copy number indicated by the red bars appears to be enriched in those cell lines which have high sensitivity to panatinib. So this is consistent with what has been reported before. For FGFR1 protein, a similar trend is observed. That is, the high protein expression is enriched in tumors in cell lines that are highly sensitive to the drug, and the expression is much lower in those resistant to the drug. Likewise, the FGFR1 R1 mRNA showed a similar pattern. That is, the expression is much higher in the sensitive cell lines versus the resistant cell lines. If you plot all the 3 biomarkers in a ROC curve, it actually shows the gene copy number is a very modest predictor of drug sensitivity, as shown, indicated by a AUC value of 0.691. In contrast, the FGFR1 mRNA or protein have much better predictive power. It has AUC values about 0.9. So, as indicated, at least in the cell line studies, the RNA and protein of FGFR1 appears to be much more predictive for drug sensitivity than gene copy number. So, in summary, the paper, the study shows that FGFR1 mRNA levels actually do not correlate with gene copy numbers. And there are cases that gene is amplified, but there's no overexpression of the RNA or protein. And the overexpression of mRNA does not involve gene amplification in many cases. And at least in cell line studies, the FGFR1 mRNA appears to be a much better predictor of drug sensitivity than gene copy number. Importantly, the RNAscope assay was able to identify a lot more patients that may benefit from therapy than gene copy numbers. In this cohort, 29% patients are positive by mRNA versus only 8.6% positive by gene copy numbers. And this may be important since RNAscope assay may be able to increase response rate in a clinical study by excluding DNA amplified but overexpressing tumors from a trial. So all of these results have prompted a phase 2 clinical trial, which is in progress right now, that will formally assess the predictive power of the gene copy number, or mRNA, as a biomarker for ponatinib. To summarize all the 4 publications I just discussed, the first one used the RNAscope to discover 2 subtypes of head and neck squamous cell carcinoma with different modes of Heregulin signaling. The second paper used RNAscope together with IHC to demonstrate a novel paracrine signaling mode of hedgehog signaling in gliomas, which really highlights the intratumor heterogeneity. That is, there are 2 different populations of tumor cells. One generates the hedgehog, one responds to hedgehog. The third paper used RNAscope to develop a new diagnostic assay for FGF23 for diagnosing the rare tumors called a PMT. Last paper used the RNAscope to demonstrate that FGFR1 mRNA to be a superior biomarker to a gene copy number as a potential companion diagnostic for lung cancer. So these 4 publications not only 4 out of over 100 peer-reviewed publications by users of RNAscope technology in the last couple of years. And as you can see here, there has been a very rapid growth in publications that uses RNAscope technology. And these publications included many high-profile journals like Nature, Cell, Science, and etc. To summarize, we can see that the single molecule sensitivity and high specificity of the RNAscope assay can be readily translated into benefits in cancer research. That is, the ability to detect very low levels of transcripts or biomarkers. The single cell resolution and quantification allows the interrogation of the tumor microenvironment, allows to reveal the underlying intratumoral heterogeneity. The assay is universal. It can be designed for any new markers you identify. It's not a problem if you don't have antibody for the markers you just discovered. An RNAscope assay can be designed in 2 weeks if it's not already on our catalog. The assay can be run in different formats, different color choices to fit your experimental design, and can be also run either manually or automated. And this should really speed up your validation of your biomarker studies and accelerate the translation of your discoveries into clinical applications. I think this is the end of this part of the talk. I will stop here and try to answer some of your questions you may have. What question you have.
Thanks, Jordan. I think we already have the first question here.
Okay.
So this person is using GEM mice where they have targeted deletion of our gene of interest to the prostate, but don't see difference in expression level between a wild type and our knockdown by IHC PCR-ish. Would this be a potential target for RNAscope?
Okay, I'm sorry. I'm trying to scroll through the questions on the box in the box. Okay, I don't see the question. Okay, can you repeat the question again, Claudia? Sorry.
Okay, I'll just read it out. So thank you for your talk. We are using tissue from GEM mice where we have targeted deletion of our gene of interest to the prostate, but don't see difference in expression level between WT, so wild type, and our knockdown by IHC PCR-ish. Could RNAscope be used for this?
Yes, absolutely. We actually have multiple publications that uses the RNAscope technology as a way to verify the knockdown or knockout, especially when you have a conditional knockout that occurs in a specific tissue. Yeah, this can be very, very effective. Yeah.
Okay, she's saying thank you.
Yeah. Okay, looks like there are a few other okay, so one question is, how is the RNAscope different from a light microscope or IF scope? Is quantification intensity based? Okay, so this the question is maybe related to the fact that we named our product as a scope. So in fact, our product is not a scope, is not a microscope. Is basically a technology that generates stained slides that can be visualized under a microscope. The quantification of RNAscope staining slides is actually not purely intensity based, although it's kind of related to intensity. As I mentioned during the talk, that the best way to quantify RNAscope staining slide is by counting the number of signal dots on a cell-by-cell basis. Of course, when you have more dots per cell, the signal will appear more intense. But in some sense, that really the number of dots is more relevant than the intensity of the individual dots. Okay, I hope that answers that question. What resolution answer? Right. Okay, any more questions?
So I don't see any more here. Do you have any more on your side, Jean?
Yeah, I don't see. No, I think that's all.
Okay. So if you have any more questions, please don't hesitate to contact us. And you also can find further information, especially on biomarker research and cancer research, on our website. So it's www.acdbio.com. There's also our complete publication list. So all the 120 publications we have during the last years is downloadable there. And you also will be able to have a look if your probe of interest is already on the shelf, and as Jojon said, if not, we are happy to create it for you within 2 weeks. So again, thanks everybody for your time today, and yeah, we hope to see you at our next webinar. Thank you.
Thank you. Bye-bye.